I want to introduce our second speaker, Dr. Viviana Agus. Viviana has more than 10 years of experience in the development of cellular assays and their adaptation to high-throughput screening, mostly using fluorescent and luminescent readouts on the FLIPR instrument. Her main experience is with ion channel cell line development and characterization. She has been fully dedicated to optogenetics projects for over 1 year, focusing on the development of both light actuators and light sensor cell lines suitable for high-throughput screening. And with that, let's turn it over and start Viviana's presentation.
Welcome, everybody. This webinar will be about optogenetics and cellular assays, the revolutionary use of light to drive high-throughput screening. My presentation will include different topics. I will start making a brief introduction on optogenetic technologies, giving an overview of all the potentialities of this technique, and introducing the fundamental concepts of light actuator and light sensor. I will then present the results that we obtained in the development of cellular assays for different optical tools, a Channelrhodopsin, a bPAC enzyme, a voltage sensor, and a calcium sensor. And then I will conclude summarizing the main achievements of our work and the possible future applications. The word optogenetics describes a technique which uses genetically encoded elements which properties are modulated through light. These two aspects deliver the concepts of spatial resolution and temporal resolution, respectively. If you use the light to control a cellular process, this means that you can control the duration and the frequency of your target stimulation. Basically, you can be reversible, switching on and off your specific activity. And being the light and electromagnetic waves, you can use different wavelengths to run multiparametric experiments and to control different processes. Moreover, the genetic component of your optogenetic tool will allow you to act only on the specific light control pathway of your cell line or cell compartment, avoiding a lot of off-target effects and toxicity issues in a very cheap and not invasive way. And all these aspects are very advantageous for biological investigations because they are definitely something you cannot reach with a classical drug application or even with electrical stimulation. Just to give you an overview of which are all the optogenetic tools that can be potentially applied to drug discovery, let me introduce the concepts of light actuator and light sensor. Light actuators are proteins which activity is induced by light stimulation. For example, we have light-activated ion channels, the so-called Channelrhodopsins. We have light-activated enzymes, G-protein couple receptors, and also mechanisms of protein-protein interaction or translocations, which can be triggered by a light stimulus. On the other side, light sensors are genetically encoded elements which change their light emission upon a change in a cellular parameter. For example, we have the fluorescent voltage indicators or the very well-known fluorescent calcium indicators, such as the GCAM sensors. So in general, the use of light actuators and light sensor would allow you to obtain a precise stimulation and measurement of many different cellular pathways. Some considerations should be done when using optogenetic tools in HTS. First of all, a good expression level might be difficult to obtain as many of these optogenetic tools derive from unicellular organisms. Then your optical tool should have good light sensitivity, as many HTS readers have very weak light sources. And then the spectral properties of your tool should fit the optics of your HTS reader. And finally, everything should be robust and miniaturizable. The reasons why we decide to apply optogenetic technologies to HTS are the need to develop more precise assays where the use of light can be much more temporally precise and target-specific than the use of chemicals. Moreover, light control assays can combine biological relevance with cost-saving needs, representing a promising alternative to high-throughput electrophysiology techniques. The use of light can be also less invasive and more physiological than other methods, generating much less of target effects. So in summary, we thought that investing in this innovative technology would definitely be beneficial to the exploration of the future of high-throughput screening. In particular, we investigated the possibility of running optogenetic projects in an HTS-compatible fashion using a 384 well plate format and the fluorescence plate imager instrumentation, the FLIPR system. Optogenetics activities started in AXXAM in 2013 with the first project on Channelrhodopsin 2 ion channel. We then briefly validated the light-gated bPAC enzyme, and then we started working on voltage sensor and calcium sensor cell lines. For all these projects, we stably expressed optimized nucleotide sequences in heterologous recombinant systems. We selected functional pure clones at the FLIPR, then we validated ion channel targets also by electrophysiology. The first explorative project that we ran was a Channelrhodopsin. This protein derives from a unicellular algae ispot, the Chlamydomonas reinhardti. It is a very well-validated system where the blue light stimulation of Channelrhodopsin can rapidly induce a cation influx and a depolarization of the plasma membrane of the host cell. We decided to use a mutant version of the Channelrhodopsin 2 because it showed an improved photocurrent and light sensitivity compared to the wild-type version, even if with a much delayed kinetics. So even if the use of a faster Channelrhodopsin would be the ideal system to deliver precise electrical pulses to the cells, we decided to start with this delayed mutant in order to produce the first proof of feasibility that optogenetics can be applied to HTS. We use the Channelrhodopsin mutant to induce a sustained and prolonged membrane depolarization and also to consequently trigger the activity of a Ca1.3 voltage-gated calcium channel. So with this first explorative project, we aim to determine if the FLIPR light sources have enough power and suitable wavelength to modulate the Channelrhodopsin 2. If the Channelrhodopsin photocurrent would be strong enough to activate and modulate the Ca1.3 channel, and if all these processes can be run in a 384-well format. To configure the optogenetic assay for Ca1.3, stably transfected the 3 subunits of the Ca1.3 voltage-gated calcium channel in HEC cells, expressing the ChR2 potassium channel. This channel maintains the resting membrane voltage to very hyperpolarized values, keeping the Ca1.3 channel in a closed state, and also allows the modulation of membrane voltage upon application of different concentrations of extracellular potassium. In this cell line, we stably express the light-gated Channelrhodopsin 2 mutant, which can trigger cation influx upon light stimulation, and can thus be used to modulate the membrane voltage and the Ca1.3 channel state in an optogenetic manner. In the end, we optimized an optogenetic protocol to identify state-dependent blockers of Ca1.3, meaning therapeutically relevant compounds which act more potently on the inactivated pathological state of the Ca1.3 channel. And due to the important potentialities of these optogenetic tools, AXXAM decided to acquire the license to use Channelrhodopsin mutants according to the published patent documents. The FLIPR validation of this assay included 2 different steps. The first was to demonstrate that the FLIPR optics was able to stimulate the Channelrhodopsin photocurrent and to induce a consistent membrane depolarization. So we delivered blue light stimulation and we measured the membrane voltage changes with a fluorescent membrane potential sensitive dye. As shown in these kinetic traces, the blue light stimulation induces strong and sustained membrane depolarization, which was progressively recovered following Channelrhodopsin deactivation. If we measured the dye fluorescence over time, we could demonstrate that after about 10 minutes from the first blue light pulse, the membrane voltage recovered about 50% of its initial value, and after 30 minutes, it's completely repolarized. So after demonstrating that the Channelrhodopsin is well functional and can be well activated by the FLIPR blue LEDs, we sought to determine if this blue light stimulation would induce a calcium influx to the Ca1.3 channel. As shown in this graph, the first blue light stimulation induced a very nice and robust calcium influx, as reported by a fluorescent calcium-sensitive dye. Due to the sustained membrane depolarization induced by the Channelrhodopsin, if the Ca1.3 cells receive a second pulse of blue light, part of the channels will be still in the non-conducting inactivated state due to the elevated membrane voltage value. Following the membrane voltage recovery kinetics, after about 10 minutes from the first blue light pulse, the Ca1.3 calcium signal was about 50% of its initial value, suggesting that 50% of the Ca1.3 channels are still in the inactivated state. So we use this condition of repetitive light stimulation to study the Ca1.3 channel in a resting state during the first stimulation or in an inactivated state when we pulse after 10 minutes. We can very well validate our light protocol by using known state-dependent blockers of Ca1.3, such as the isradipine compound. The pharmacology obtained with the repetitive light protocol was very well consistent with literature data and also with patch clamp experiments where channel states are modulated using different holding potentials. The signal obtained with the light protocol showed robustness and performance definitely comparable with the classical protocol of membrane voltage modulation by potassium challenges, thus fulfilling all the requirements for a reliable and miniaturizable cellular assay. So the conclusions of this first Channelrhodopsin project are that the development of a precise and high throughput stimulation of electric activity of cells is potentially feasible, and this is really the main relevant message. Besides that, we were able to develop a robust HTS-compatible assay to study carbon-3 dependent blockers in an optogenetic way. And this tells us that other Channelrhodopsin proteins with faster kinetic features might be important future projects to achieve a precise ion channel modulation in a fully HTS format. So after the success of the first Channelrhodopsin ion channel project, we investigated the second family of optogenetic tools, the bPAC enzyme. This protein is from vegetable bacterium and it encodes for a blue light activated adenylyl cyclase, so it converts ATP in cyclic AMP. And due to the importance of the CAM second messenger in regulating many cellular processes, we decided to set up a recombinant assay in which the blue light stimulation of bPAC induces the production of cyclic AMP in order to demonstrate if the FLIPR light sources has suitable energy and spectral properties to activate also this optogenetic tool, and also to see if the CAM produced by the bPAC enzyme would be suitable to induce the activation of a CNG channel, HCN2. We set up the optogenetic assay for HCN2 stably transfecting HEC cells with HCN2 CNG channel and with the bPAC enzyme. Fast-screen application was used to stimulate endogenous adenylyl cyclases and to induce CAM production in a classical way, while blue light pulses applied with the FLIPR were used to stimulate CAM production by bPAC in an optogenetic way. As shown in the kinetic traces, the blue light stimulation produced a fast and robust membrane depolarization as reported by a fluorescent membrane potential sensitive dye, and this depolarization can be specifically inhibited by the ZP7288 compound, which is a known HCN2 channel blocker. If we compare the classical force-pulling protocol with the optogenetic bPAC stimulation, we can see that bPAC is delivering a very similar robustness and assay reliability, thus fully validating the possible application of this optogenetic tool to the HTS process. So in summary, we demonstrated that the levels of intracellular cyclic AMP can be regulated optogenetically using bPAC enzyme. This might be useful to obtain a reversible modulation of CAMP levels with less off-target effects compared to fast-screen stimulation, but with similar robustness and assay reliability. We could obtain a nice stimulation of bPAC enzyme by FLIPR blue LEDs, and we could set up an optogenetic assay for HCN2 using bPAC optical tool. So these results open the possibility of using bPAC to optically modulate other CAMP-regulated processes, and this might deliver to the HTS process significant improvements. In parallel with the validation of optogenetic light actuators, such as the Channelrhodopsins and the bPAC enzyme, we decided to validate the use of optogenetic light sensors to measure in a precise and robust way complex electrical signals. In particular, we focused our attention on Arclight voltage indicator. This protein is composed by the voltage sensing domain of Siona intestinalis phosphatase fused to a fluorescent domain, the superecliptic pH fluorine. This genetically encoded voltage indicator has been widely used for both in vitro and in vivo studies, and its big success is due to the fact that apart from being less toxic and more photostable than organic dyes, it possesses very good light sensitivity, and it is the state-of-the-art best compromise between brightness and speed. The response of Arclight to membrane voltage changes is about 40% of fluorescence quench upon a plus 100 millivolt membrane depolarization, with response kinetics in the range of 10–50 milliseconds. These features combine an improved speed compared to optional dyes, which respond with a time scale of seconds, with an improved brightness compared to potentiometric dyes. So with Arclight, one can measure fast signals, which wouldn't be detectable by the slow optional dyes, and with good fluorescence response, which would be much smaller with potentiometric dyes. In order to validate the potentialities of this voltage sensor, we generated stable clones, where the Arclight indicator is used to detect the activity of a potassium, a sodium, or a chloride channel. As a first experiment to demonstrate if the Arclight sensor can be well expressed in HEK cells, and if its fluorescence can be well detected by the FLIPR optics, we co-express the Arclight sensor together with the inward rectifier potassium channel ChR2 .3. In this cell line, it is very easy to obtain a huge membrane depolarization by the application of high extracellular potassium. We obtain a very nice and dose-dependent fluorescence quench of Arclight when injecting increasing concentration of potassium at the FLIPR. This confirmed us that the FLIPR optics is suitable to detect the Arclight voltage sensor signal, and the obtained response of 40% of fluorescence quench was pretty in line with literature values. Arclight sensor was dose-dependently detecting also membrane hyperpolarization induced by a switch from high to low potassium solution. The Arclight cells observed at the microscope showed the bright, homogeneous, and plasma membrane localized fluorescence with no apparent toxicity effects. In current lamp experiments here depicted in green, you can see that the Arclight expression didn't alter the biophysics of ChR2 cell line. Also, the fluorescence changes of Arclight, here depicted in blue, are linear over physiological ranges of voltage changes induced by changes in potassium concentration. The last but very important feature of Arclight is its spectral purity. In fact, the sensor fluorescence is very well detected by the blue-green LED filter set, but is totally dark if measured with a yellow-orange LED filter set. And this is a very valuable feature because it would leave the other wavelengths free to be used to simulate other optical tools or to measure other parameters, so more in general to run multicolor, multiparametric experiments. After the first successful validation of Arclight with the ChR2 potassium channel, we decided to fully validate this sensor with different ion channel targets. We expressed Arclight sensor with now 1.5 sodium channel, and we could get a nice and dose-dependent fluorescence quench upon the application of increasing concentration of ratidine opener, and this Arclight quench could be dose-dependently reverted by the pretreatment with amitriptyline blocker, confirming that the now 1.5 sodium channel pharmacology can be studied in a consistent way using the Arclight indicator. We use Arclight also to detect the activity of TMM16A chloride channel. As shown in the FLIPR trace, the injection of ionomycin stimulus induced a strong Arclight fluorescence quench due to the chloride ions efflux through TMM channel, and this was potently and specifically inhibited by the niflumic acid blocker. Finally, we tested the response of acid channel by the use of Arclight indicator, demonstrating a nice Arclight fluorescence quench upon mid-toxin stimulation, which can be well inhibited by diminazate aceturate acid blocker. So in summary, the Arclight voltage indicator displays a lot of important features that would be very attractive for the detection of fast voltage changes in a robust way, as summarized in this list. What we could successfully demonstrate is that the Arclight sensor stably expressed in HEK cells can be very well measured with the FLIPR optics, and this is one of the most important aspects. Moreover, we developed stable pure clones where Arclight sensor robustly detects different ion channels activity. And the future possibility to use Arclight to detect fast and reversible electrical signal in an HTS-compatible way are the more valuable aspect of our work. And thanks to the nice spectral purity of Arclight, we decided to validate this sensor in combination with a red fluorescent calcium sensor in order to set up multicolor, multiparametric experiments for the simultaneous detection of voltage and calcium changes. We use the AXXAM genetically encoded calcium sensor based on the MRuby fluorescence protein, which senses increasing intracellular calcium as increase of orange fluorescence. We stably expressed the AXXAM sensor in HEK cells, and we could demonstrate that the FLIPR optics is well suitable to detect the calcium signals elicited by endogenous muscarinic receptor agonist carbachol or by the calcium ionophore ionomycin. The cells showed a nice and cytosol-located red fluorescence when observed at the microscope. We also co-expressed AXXAM sensor with Arclight, and we could demonstrate that a dual fluorescence protocol can be run at the FLIPR to simultaneously detect in the same well the calcium changes measured with AXXAM together with the voltage changes measured with Arclight, thanks to the nice spectral separation of these two indicators. So these results really demonstrated that a multicolor, multiparametric analysis can be well done in an HTS-compatible format, but they use only of genetically encoded indicators with no need to use any fluorescent dye, and thus making the screening process easier, cheaper, less toxic, and also definitely more precise from both a spatial and a temporal point of view. So to summarize the main messages of my talk, we started from the understanding that optogenetics techniques has big potentialities in many fields of biological investigation, and also that the spatial and temporal precision of this technique can increase the quality of HTS while reducing the cost. By the use of both light actuators or light sensors to modulate and measure biological processes in a precise and more physiological way, optogenetics might elevate so much the content and overall quality of HTS process, opening new unexplored possibilities to identify active drugs. So our work showed that different optogenetic tools can be well expressed in recombinant systems, showing good functional data also in stable pure clones. And the most interesting result is also the fact that the 384-well HTS-compatible reader, like the FLIPR imaging system, can be used to run optogenetic experiments using its optics to both stimulate light actuators or detect light sensor activity. So in summary, we demonstrate that optogenetics can meet HTS, and AXXAM is continuing investing in this field, and we are currently validating other optogenetic tools to fully explore the powerful potentialities of this technique. And after saying that, let me thank all the AXXAM people who collaborated in the realization of this project, and of course I'm glad to answer to any other questions, and thank you very much for attending this webinar.